Welcome to the Alg-AD exhibition, located on the experimental farm of Découvrez in the west of France. I have the pleasure to be your guide throughout this visit. We will discover together the links between anaerobic digestion and microalgae and the potential of these microorganisms in the feeding of livestock. I propose to start with the topic of anaerobic digestion, which is at the heart of the Alg-AD project. The anaerobic digestion is a natural biological process of degradation of organic matter by bacteria in an environment deprived of oxygen. In the wild, the reaction can occur, for example, in marshes. When it is induced by humans, it takes place in a controlled environment, in sealed tanks called digesters. Many biological wastes are fermentable and can be used in anaerobic digestion, such as manure, slurry, or other livestock effluents, crop or food waste. The molecules degraded by this process are transformed into biogas, a mixture composed mainly of methane and carbon dioxide that can be recovered through a cogeneration plant in the form of electricity and heat. When injected into the natural gas network after purification, we speak of biomethane. In Europe, there are nearly 17,800 anaerobic digestion units that produce 65,000 gigawatts of electricity and more than 19,000 gigawatts of biomethane each year. From the anaerobic digestion process, a nutrient-rich residue called digestate remains. It can be separated into two phases, a liquid phase rich in nitrogen, which is of particular interest to Alg-AD researchers, and a solid phase rich in phosphorus. Each year, 180 million tons of digestate are produced in Europe, the vast majority of which is used as fertilizer for agricultural plots. But in some regions, its spreading is limited due to the vulnerability of the environment to nitrates. It is in this context that the Alg-AD project explores new market opportunities for digestate. The project researchers are studying the cultivation of microalgae on different types of digestate in order to produce biomass that can be used in animal feed. Microalgae have proven to be excellent candidates to achieve these goals. Microalgae are unicellular and mostly photosynthetic organisms that appeared on our Earth billions of years ago. Today, they are responsible for almost half of the Earth's oxygen, while representing only 1% of the biomass of photosynthetic organisms, and they sequester almost half of the CO2. In aquatic environments, they are also very important, since they are the first link in the food chain. Endowed with an exceptional capacity of adaptation, they can develop in most aquatic environments, even under extreme conditions. There are hundreds of thousands of species, but to date, only a few dozen species are used on an industrial scale. However, their applications are numerous. Living, they can clean up the air or purify wastewater. Cultivated in biofacades, they could improve the insulation of buildings while consuming the CO2 they emit. But their greatest potential lies in their transformation biorefinery processes in order to exploit their great wealth of lipids, proteins, and other molecules of interest. Biorefinery is a technique that allows the extraction of high added value molecules, such as pigments, antioxidants, or omega-3, that are of interest to many industrial sectors, in agriculture as well as for livestock feed, as biofertilizer or biofuel, but also in cosmetics and pharmaceuticals, or even as a food supplement for human nutrition. They are currently mainly used in aquaculture for the rearing of mollusks and fish larvae, but their nutritional qualities and the absence of competition with agricultural land use make them excellent candidates for feeding other livestock. The culture of microalgae remains delicate because all the species do not have the same physical and chemical needs. Some are capable of photosynthesis to produce the organic matter necessary for their development. They are called autotrophic organisms. Others are able to grow in the absence of light, but they require a source of external organic carbon to produce energy. They are called heterotrophs. Others even have a metabolism called mixotrophic, that is to say, able to function in autotrophy or heterotrophy. On an industrial scale, they can be cultivated in open ponds or in closed tubular structures called bioreactors. The Alg-AD project has opted for the second solution because it allows to limit the risk of contamination of the cultures while enabling a fine control of the parameters of growth, like light, pH, temperature or agitation. Now I would like to tell you more about the activities of the Alg-AD project. Alg-AD is a European project that brings together 11 partners from the United Kingdom, France, Belgium and Germany. It is financed at 60% by the European Regional Development Fund, within the framework of the Interreg Northwest Europe programme. The Alg-AD project presented on this site has been designed with a circular economy logic. In practice, as we have seen, livestock effluents, crop or food waste are collected and then processed in AD units to produce biogas, energy and heat, and finally digestate. This digestate will be separated into two phases, solid and liquid. The latter, rich in nitrogen, will be used as a substrate for the cultivation of microalgae, which in turn will be transformed into feed for livestock. The loop is closed. To carry out these activities, 3 pilot sites have been deployed in the UK, France and Belgium. Each of these sites is studying different strains of microalgae grown on different types of digestate under different conditions. The English site, located in Plymouth, is associated with the Langage AD anaerobic digestion unit and the University of Swansea. There, the microalgae Chlorella vulgaris and Cynodesmus obliquus are studied. They are cultivated in autotrophy, then in mixotrophy in a 5,000-litre photobioreactor. The Lamballe site in France, the microorganism Aurantiochytrium mangrovi is grown heterotrophically, in the dark in a 2,000-litre bioreactor. A supplementation of glucose, yeast extracts and peptone is necessary to ensure the growth of the organisms. The CNRS and its laboratory Limar, the University of Western Brittany and the COPERL are collaborating on the site. Finally, on the Belgian site, located near Ghent, InnoLab and the University of Ghent are cultivating in autotrophy a consortium of 2 microalgae, Chlorella and Desmodesmus, equipped with 2 photobioreactors, one horizontal, the other vertical, for a total volume of 3,000 litres. In order to be used as a culture substrate, the digestates must undergo a pre-treatment to eliminate pathogenic organisms, suspended particles, and the colour of the liquid that hinder the growth of the microalgae. For this purpose, several techniques have been tested. Ultrafiltration on 100 to 300 kilodalton membranes at the English and French sites, microfiltration on 0.2 micromembranes at Swansea University, and polypropylene bags with 10 micropores at the Belgian site. The digestate is then diluted with water, as too high a concentration of nitrogen can be toxic to the microalgae. Finally, the solution is injected into the reactors, which will be inoculated with microalgae. The cultivation time and biomass concentration at harvest vary according to the species. On our 3 sites, harvesting occurs between 2 and 21 days after the beginning of the culture, and the concentrations obtained range from 1.5 to 40 grams per litre depending on the species. After harvesting, the algal biomass, rich in proteins and lipids, is transformed in order to be incorporated in animal feed. It is via refinery processes such as enzymatic hydrolysis used in the Alg-AD project that our teams are able to extract high added value molecules from microalgae for animal feed. If you want to know more about the results of our pilot sites, the biorefinery processes, the cultivated microalgae and the actors of the project, I invite you to visit the stands of our partners or to visit our YouTube channel to follow the news of the project. Thank you for all your participation to this visit,